Hi guys, I'm Nick and I'm one of your SI leaders for Biology 5A. And today in this video, we're just going to quickly go over a few things regarding crosses. So we've been looking at these in lecture for a while now, at least the past few, and yeah, we're just going to go over them really quickly. So what we're looking at here is a monohybrid cross. Now when we say mono, you'll notice that there's still two different things. So in a monohybrid cross, we are looking at the variation. We have our two different alleles. We have our dominant allele, which can be defined as big A, and our recessive allele that can be defined as little a. But we're looking at one trait here, right? Because both of these, in turn, represent two variants of one trait. So in a monohybrid cross, we're going to cross, let's say, this organism's alleles for the trait with this one's. So we have a heterozygous up top and a heterozygous on the side, and this is a Punnett Square. Right, so all we're going to do is in each of these boxes, we're going to look at what two things make up what goes in the box, and then we're going to put it in there, right? So when we cross two heterozygous individuals, what we actually end up with is a ratio of 3 to 1. You'll notice that even though these are heterozygous, they're going to display the dominant phenotype because they have at least one dominant in them. So our 3 to 1 ratio is essentially we have 3 offspring that will show the dominant phenotype of this trait, and we will have one that shows the recessive. And that's just in a monohybrid cross, right? We're only looking at one trait here. So now going forward, when we look at a dihybrid cross, we're looking at two things that could possibly be linked, right? So let's say we're looking at two organisms here, one that has all dominant and one that has all recessive. So from this, what we can actually see is there's a huge mixture of things that can arise from this, right? If we look at our dominant ones, we can have offspring that may have a big A and a big B in them. And same thing, we can look at ones that are heterozygous with the dominant one being the A, but the B being the recessive one. And we can have it vice versa, where the A is the recessive one and the B is the dominant one, or we could have them showing both recessive. So because we're looking at two traits here, and we're looking at a dominant and recessive allele for each one, each of these different things will show two different traits instead of our monohybrid cross over here, where we'll either have a dominant if there's a big A at all, and it's going to be recessive if there's a little a at all. When we're looking at the dihybrid cross, we could have a dominant for both of the traits that we're looking at, or we could have a dominant and a recessive and a dominant, or a recessive and a recessive. And since A and B are two unique traits that we're looking at, each of the offspring will have a different product. But if we were to do the same type of ratio, where we look at the ratio of, let's say, dominant A's, and we keep going through there, we're looking for our dominant A's, and we're looking at our dominant B's, our recessive A's, our recessive B's, we end up with a 9:3:3:1 ratio. 9 to 3 to 3 to 1. All right, so going forward, last few things we need to look at are the reciprocal cross. So the reciprocal cross is essentially a cross in which we have a wild type of something. The wild type could be quote unquote the normal for that, the normal trait of whatever organism we're looking at. And then we'll have a mutant, and the mutant will have the abnormal, right? So what the reciprocal cross does is it takes a male and a female of each in these two categories, and we do two different crosses of them, right? So in the first one, we're going to take a male wild type, a normal male, and a female mutant and cross them and see what we get as the offspring. And in the second cross, we're going to take a male mutant and a female wild type and cross them and see what we get. And so let's switch over to the slides real quick. The reason we actually do this is because it helps us figure out the linkage, quote unquote, to either males or females, right? So if we look at this example over here, right, let's say our wild type, as it's listed over here, right, dominant allele, is going to be the red eyes in the fly. right? And then we'll say the recessive allele, or the mutant, is going to be the white eye, right? So our dominant, our wild type, is placed as a plus, and our mutant just has the W for white, right? So if we look at these, now we're looking at not just like in the previous examples where we were looking at A or B. Now over here, we're looking at XX versus XY, right? So our XX is going to be our female, right? In any case, we're looking at XX versus XY just based on chromosomes. Those indicate male or female. Two X's, female, XY, male. So when we're looking at this, we have our female that has two dominant alleles, right? It has an X dominant and an X dominant. And then if we look at our male, it has an XYI and just the Y, right? The reason the Y doesn't have anything linked to it is because this specific trait is what we call an X-linked trait. It is determined in the fly, this phenotype of either red or white is determined purely off of what is displayed on the X chromosome, right? So if we look at this, if the dominant allele is the red eye, had we had this X plus and an XW, so had we had a plus over here, a dominant, and a W, which is recessive, it would still display the red in much the same way that if we had a big A and a little a, it would still display whatever the phenotype of the big A is. That's essentially a sex-linked heterozygote, right? Over here we have a homozygote, homozygous dominant, right, on this X-linked trait. And over here we have a recessive X-linked trait, right? Because only one X is needed in the males. Since males only have the one X, only the one is needed to show this white trait. So when we cross these two, the reason there's only one over here is because essentially the ratio is going to be the same, right? If these two are different, we can list both of them, but these two are the same. So had we put another box underneath, right, we would have been looking at the same 50-50 split anyways, right? So if we look down here, right, we have our XX, which is going to be our female, and like we just said, we have a plus over here and a W over here, right? But because the plus is the dominant allele, as defined up here, and the W is the recessive allele, this X plus XW is going to display the red eyes, right? And then when we look over here, we're going to have an X plus with a Y, right? This indicates our male, and because we see the plus, it's going to have a dominant allele, and it's going to display the red phenotype. So this is one of our crosses. Now if we cross the white-eyed female, XW with the red-eyed male, X plus Y, right? Over here, we still only need one line because XW is going to appear in both of them. Over here, we see an X plus XW, indicating another heterozygote for the females. While over here, we see an XWY, which indicates a mutant for the males. So our XX is still going to display the red, while the XWY is going to display the white. And these are going to be the F1s of each reciprocal cross, right? We're crossing a female with the wild type with a male mutant, and then over here we're crossing a female mutant with the male wild type. Hope this video helped, and we really appreciate you guys coming to our SI sessions. If you guys have any further questions, make sure to leave a comment, and all of our times and Zoom links should be listed in the description. Thank you for watching.